when you take the PCR when compared to the gene cloning, the objective here or the most important features of PCR when compared to gene cloning is there is selective amplification. Of the gene of interest. Be it a flowering gene or a nitrogen fixing gene or a high efficiency photosynthetic gene, and amplification occurs in vitro. If you remember, it occurs in a PCR machine. And what are the requirements? The requirements for the polymerase chain reaction are the presence of tag polymerase, which is a highly thermostable enzyme. Derived from Thermus aquaticus. Derived from Thermus aquaticus. Then again, in addition to the tag polymerase, we also require RNA primers. We require 2 RNA primers. Then again, we require free D NTPs, free nucleotides, free nucleotides. The amount of DNA which is required, is primarily given as in nanograms, very small amounts. Nanograms of the DNA is required for a PCR. The time taken is 4 hours max. Within 4 hours, we can make billion copies of your sort of a DNA, and it is an automation technique. As implied, it has wider applications when compared to gene cloning. It has wider applications in agriculture, medicine, diagnostics, and in relation to environment and conservation. The applications of PCR are extremely wide. Now, a very important point for you to understand is the time, use of DNA, as well as the different sort of requirements, and you are very specific with relation to gene of interest. So, if we are looking in terms of quality, we are looking in terms of quality, in terms of human welfare, or improving the quality of life as per the EFB definition of biotechnology, use of PCR has got myriads of applications in biology.